Good afternoon, and welcome to Life Sciences Conference on targeting the oncogene, the eukaryotic initiation factor 4E at the bench and in the clinic. I'd just like to say that I'll be taking all questions at the end, so just you can type them when you like, but I'll be answering them all at the end. And if you have any further questions, or if you're watching the recording, my email is on the bottom of this slide, so you can go ahead and email me any questions that you might have. So let me tell you a little bit about the eukaryotic translation initiation factor 4E, which I'm going to just nickname 4E here. EIF4E overexpression leads to transformation in cell culture in animal models and also rescues cells from apoptotic stimuli. 4E is elevated in 30% of human cancers, and its overexpression in cells can cause, as I said, tumors, and also 4E overexpressing cells have appeared to develop an oncogene addiction to EIF4E, providing a therapeutic window for potentially targeting this protein, as you'll see later on in this talk. EIF4E is elevated in approximately 30% of human cancers. These include acute myeloid leukemia, blast crisis, chronic myelogenous leukemias, as well as a broad array of different other types of leukemias. Note that some leukemias have a very high penetrance of high 4E, such as head and neck cancer, whereas where 98% of head and neck cancers have elevated 4E, whereas other cancers like breast cancer, it's a fraction in specific subtypes that have elevated 4E. Now, a basic question clearly is, how does EIF4E become elevated? And there's a wide variety of ways this can happen, and I've only listed 3 here, but there's undoubtedly many more. Increased stability of 4E RNA by a factor called HUR is one way that 4E levels become elevated in cancer. Gene amplification is another way that 4E becomes elevated in cancer, and increased transcription, particularly noting the NF-kappa B and c-MYC pathways for elevating EIF4E levels. And I'd just like to note that that these multiple pathways here can work together. So for instance, we can see increased RNA stability and increased transcription in the same cells. So I just like to go through what are the effects of EIF4E. Many of you will be familiar with 4E as a generalized translation factor. And I want to take a moment to show you that EIF4E's effects are specific. For instance, many years ago, in Kaufman et al., we can see using S35 methionine that EIF4E is causing S35 methionine, not causing global changes in protein translation, whereas we can look at another picture where we can see that EIF4E is actually antisense oligonucleotide treatment to 4E does not lead to an overall change in global translation, but yet it has a specific effect on translation, as shown by the studies of Jeremy Graf et al. I'm just having a minor technical issue with my slide deck for a minute, so just give me a second while I try to get my deck to work again. But I think it's a very important point that it's known that 4E is actually involved in only specific effects in translation and in and not general effects. Now, the other thing that many of you will be familiar with is the idea, as I said before, that 4E is a global translation factor, but it turns out that 4E also plays an important role in the nucleus, in the specific RNA export of a subset of translation, of a subset of transcripts. And so what happens in this is that I'm still having a tremendously hard time with my web interface. I apologize. What happens with this is that 4E can actually move subcellular compartments. So it's sometimes in the nucleus and sometimes in the cytoplasm, as you can see here, where we're looking at 4E movement through the embryonic stem cell differentiation from embryonic stem cell to macrophage. And we can see here that clearly clearly the localization of 4E is completely altered in this case. And now the slide deck is completely frozen, and I don't know what to do because I can't even, okay, we'll see. Sorry, so we'll go to the next slide, where what you're seeing here is that 4E is got two main functions. As you could tell from the confocal micrograph above, we could see that 4E is elevated in the nucleus and in the cytoplasm. So in the cytoplasm, 4E plays its traditional role in translation, and it specifically targets the translation of RNAs that have complex 5' untranslated regions. And as you could see from the earlier data I showed you, not all RNAs were having their translation modulated by either 4E overexpression or by antisense oligo reduction in 4E. What we can see here is the 4E crystal structure of 4E, and we can see it binding the methyl 7 guanosine cap, shown in yellow, and together it binds to the translation machinery, allowing engagement of translation, and this contributes to the proliferation and apoptotic rescue functions and thus oncogenic transformation properties of EIF4E. Such RNAs that are involved in this particular process include genes such as vascular endothelial growth factor and ornithine decarboxylase, which have complex 5' untranslated regions, and allows us to look at, allows the specific translation of these targets. Now this side, the cytoplasmic side of the equation, many of you will already be familiar with. However, 4E, as you saw, is also found in the nucleus. And again, 4E must bind the methyl 7 guanosine cap structure in order to act in the RNA export of specific RNAs. 4E must also interact with specific recognition factors that bind an element called a 4Z element in the 3' UTR of target RNAs, which have a specific secondary structure shown here. These target transcripts have their export elevated by 4E, independent of any effect that may occur also at the translation level. We estimate approximately 700 transcripts are controlled in this way. Some of these transcripts, like ODC, are also translation targets, whereas other targets, such as cyclin D1, seem to be mainly export targets. And for instance, VEGF is a translation-only target. In order to be sensitive to 4E at both the export and translation levels, you must have a complex 5' UTR, and you must have this 3' element in UTR, the 15 nucleotide 4Z shown. You'll notice that 4E, so in this way, 4E can coordinately regulate transcripts involved in the same biochemical pathways such as proliferation and apoptotic rescue. And also note that both the nuclear and cytoplasmic functions of 4E require its ability to bind the 5' methyl 7 guanosine cap structure. So if we look at the evidence supporting an RNA export function 4E, I'm showing you three specific sets of evidence. First, we can take the nuclear fraction of cells and do an RNA immunoprecipitation, or what I'll call a RIP, and we can look at specific RNAs using quantitative PCR, such as cyclin D1, which is a 4E export target, and GAPDH, which is our negative control. And if we IP4E, we can clearly see that in cells in the RIP that we have a substantial enrichment of cyclin D1 for 4E. If we treat these same RIPs with M7-GPPG, an analog of 4E's normal ligand, we can clearly see that that we've lost the interaction, proving its cap dependence. And if we use a negative control that is not a competitor, we don't have an effect. So here you can quite clearly see cyclin D1 is a target, and in green you can quite clearly see that GAPDH is not a target, indicating this is specific. And importantly, in the IgG controls, we see no immunoprecipitation. Now, if we take our cells and fractionate them into the cytoplasmic and nuclear fractions, and look at the ratio of cytoplasmic to nuclear cyclin D1 RNA, for instance, we can see in 4E overexpressing cells, we substantially increase the export of cyclin D1, and this permits an increase in the expression of the cyclin D1 protein without having to change either transcription or translation. If we take a mutant that is inactive in promoting translation, but is active in promoting export, we can clearly see that we still have a nice export phenotype. If we take a 4E mutant incapable of binding the cap, this phenotype is gone, again reinforcing the requirement for the cap. In the final panel, we can see that if we do an RNAi experiment and get rid of 4E, that in this case, instead of looking at the nuclear to cytoplasm, the cytoplasmic to nuclear ratio, I note that we're looking at the inverse. So we're looking at the nuclear to cytoplasmic ratio, and you can clearly see that in this case, when we knock down 4E, we substantially increase nuclear retention. So all this really supports the idea that 4E binds specific RNAs in the nucleus and promotes their export. 4E, let me show you something about whether 4E is normally found in the nuclei in primary human specimens. We're going to focus today on M4, M5 AML and blast crisis CML patients because we know them to have highly elevated 4E. And as you can see here versus the normal versus the AML specimens, that 4E is very much elevated in these cells, and it's highly enriched in the nuclei of these cells. So I remind you that these are blood cells that have very fine, small cytoplasms and very large nuclei. If we look at a Western blot, where pink is normal and blue here are the primary specimens, you can clearly see that 4E and its RNA export target are highly elevated in these specimens. If we go on, we can look to see the total RNA levels by northern blot, and we can see between the patients and the normal that we don't have any alteration in cyclin D1 levels, indicating that the increase at the protein level is not transcriptional. And if we look at an assay to look at the nuclear to cytoplasmic fraction, again using northern blot in this case, and we compare a normal to leukemias, we can clearly see that in leukemias we have an increase in cyclin D1 RNA export, whereas our negative control GAPDH is unchanged. So this indicates that 4E is highly elevated in these primary AML specimens. It's highly enriched in the nucleus, and this fraction is active and able to increase the export of one of its targets, cyclin D1 RNA. And I should say that we've tested many other targets, and this is not special to cyclin D1. So if we look at the next question, how does EIF4E promote RNA export? So how does this work? So I'm only going to show you one summary slide on how this works. This is really to do with, partly to do with the ability of 4E to change the nuclear pore complex. So what we can see here is that you have in purple what we call the 4ZRNP, which is EIF4E bound to its target 4Z containing RNA, and it also binds other cofactors. And instead of traditional RNA export, 4E is exported via the CRIM1 exporter in a RAN-GTP-dependent way. What happens when you overexpress 4E is rather striking. We see a massive change in the structure of the nuclear pore complex. Specifically, you can see that the major cytoplasmic face, where the arrow is shown now, is made up of cytoplasmic fibrils in normal cells. But these fibrils are almost completely gone because the main constituent of these fibrils, RAMPP2, has been substantially lowered by 4E overexpression. We also see mislocalization of other components of the cytoplasmic face of the nuclear pore in these particular cells. And indeed, if we look at primary AML specimens, we see RAMPP2 levels are severely reduced in the AML specimens relative to the normal controls, indicating that although these experiments are mainly derived from human cell lines, they are applicable to human disease. In particular, what we see here is that EIF4E is most likely promoting cargo release, or the release of its RNA, RNP, into the cytoplasm by eliminating the steric hindrance of the presence of RAMPP2 in these 4E overexpressing cells. This permits more effective cargo release of the RNA into the cytoplasm so it can then go on and be translated, and enhances recycling and release of cargoes so that the factors that are important for export can now go back more quickly to the nucleus and allow further rounds of RNA export. So how do we clinically target EIF4E in the perspective particularly of acute myeloid leukemia? So we identified many years ago, almost 10 years ago in fact, that this small molecule, well-known antiviral drug, Ribavirin, acts as a competitive inhibitor of EIF4E. What I don't have time to show you in detail, but I'll show you a little bit, is on the following list. So Ribavirin, we will show you, directly binds EIF4E, and it directly binds in or near the overlapping with the cap binding sites. Ribavirin inhibits 4E activity in cell culture. Ribavirin 4E complexes can be found in live cells using tritiated ribavirin. Ribavirin inhibits 4E activity in patients and in primary specimens, and I'll show you our clinical trial data. And finally, loss of the Ribavirin 4E interaction correlates with clinical resistance. So this is just some data to show you the direct interaction of 4E and its ability to compete for the cap binding site. What people do to look at whether 4E can bind the cap, or one of the many ways, is to bind cap onto a methyl 7 cap cephalose. And what you see is that 4E sticks to the cephalose, as you would expect. And as you elute with cap, it comes off. And also, if you take the active metabolite of ribavirin, ribavirin triphosphate, known here as RTP, we also see this washing off. Similarly, GTP and GPPG, which are not competitive inhibitors or capable of binding 4E, do not wash off 4E from the cephalose beads, whereas you can clearly see RTP and cap do. Of course, this is using all purified protein in this assay, and so in these assays, so of course we want to know whether we can actually see these complexes in live cells, and using both experiments separately, using cross-linking IP or straight IP with tritiated ribavirin, we can see that 4E IPs substantial amounts of tritiated ribavirin versus the IgG control, indicating that ribavirin binds 4E in living cells. Again, another evidence to show that ribavirin binds 4E directly is derived from mass spectrometry. What we can do here is we had a mixture of ApoEIF4E, ribavirin, and GTP as a negative control. We only see two peaks, ApoEIF4E and the ribavirin EIF4E complexes, indicating that by mass spec, ribavirin binds 4E directly. And we see no peak for the GTP complex, indicating that GTP did not bind under these conditions, which is good as it was our negative control. We also have used NMR to look at cap binding, RTP binding, and whether it binds in the cap pocket. And so what you're looking at here is the spectral changes that occur in EIF4E upon NMR binding happen in the cap binding site. And we also do see some changes in the dorsal surface of EIF4E, which is a surface very important for all its binding partners. And similarly, cap also, the natural ligand of 4E, cap also causes similar changes onto the dorsal surface. We can see in these NMR spectra where red is ApoEIF4E and blue is RTP-bound 4E, that we see substantial perturbations in the NMR spectra, again supporting the idea that these are direct binders and they're binding, RTP is binding directly in the cap binding pocket. And I note also that RTP does not bind a cap binding mutant where the W56 is being mutated with alanine. Now, whether the effects of ribavirin, which because it's a small drug, could be highly pleiotropic, you clearly have to show that the effects that you observe on growth, for instance, are due to the presence of its effects on 4E. And so what you see is an experiment by Wilson Miller's lab, Philip Peterson in particular, where breast cancer cells were treated with siRNA to scrambled controls, or to 4E, and then treated with ribavirin. And what's very notable is that the scrambled controls, you have a nice dose-dependent effect on the repression of growth of these cells in the presence of ribavirin. In Si4E cells, despite the fact that you still have room to go, you see no further reduction in growth, indicating that the reduction in growth here is due to ribavirin's interaction with 4E. And I should also like to note that we clinically achieve well over 20 micromolar 4E with no toxicity in patients routinely. So we worked on acute myeloid leukemia because it has a very poor prognosis, and as you saw earlier, 4E levels were highly elevated, and 4E was very active in its RNA export function, which is the function that we've been interested in studying. So just to go over what the statistics currently are for acute myeloid leukemia, the majority of patients are over 60. The patients that don't receive intensive chemotherapy have an average 4-month survival. And although the CR rates are around 40 to 65%, 85% will relapse. And importantly, patients, even who achieve a CR, median survival is still only 7 months. So clearly we have a long way to go to improve the clinical outcome of this disorder. And just to drive that home, there's the following two quotes I'd like to make. Currently, management in AML in older adults, which is the majority of patients, is unsatisfactory. And the standard of care of the National Comprehensive Cancer Network guidelines recommend clinical trials as a first option for nearly all patients with AML over the age of 60. And it's also important to note that the crisis in AML is quite, well, the disease progression is quite rapid. Patients who only receive palliative care, their survival is measured in days, not weeks or months. And so this is very different than perhaps standard chemotherapy, standard solid tumor cancers that many of you may be more familiar with. So we decided to target EIF4E activity in M4-M5 AML patients using ribavirin and look to see if we could target the 4E activity and whether we could have any clinical responses. And so this first trial was a ribavirin monotherapy trial. No other chemotherapies were permitted. It was a multi-center trial between 2 Montreal hospitals, Hôpital Maisonneuve-Rosemont and the Jewish General, where the trial is coordinated and run by Dr. Sarit Assouline, and also with our collaborators at McMaster Cancer Center, in particular Brian Lieber. Our clinical responses are summarized here. 12 out of 15 evaluable patients showed clinical responses. We had a complete remission, 2 partial remissions, 3 blast responses, which is the reduction of the leukemic tumor load by 50%, 6 stable diseases, and 3 patients that didn't respond. We also saw regression of skin lesions in one of the PR patients, which had a leukemic leukemia cutis, indicating that we could target outside of the blood compartment. All our patients were very poor cytogenetics, bar 1, and had intermediate to adverse risk and unfavorable molecular markers. And all were refractory or relapsed. So what we can see here is very striking. This is the result of patient 11 who achieved a complete remission. You can see here the highly enriched nuclear EIF4E that was characteristic of the primary AML specimens we received from banks I showed you earlier. And what we also see here is that at 28 days, when this patient first achieved relapse with achieved remission with oral ribavirin, that 4E localization has changed dramatically and looks much more like a normal cell. And this correlates with bone marrow smears here that are clearly highly leukemic, and here the cells, you can see nice signs of differentiation. So this all indicated to us that ribavirin could both target EI4E activity and that we could see clinical benefit. So we looked at length with molecular response. Obviously, in clinical trials, you don't always have all the material you'd like, so we looked at the material we had, and we saw very strikingly that we were hitting 4E targets, where ribavirin after 28 days in the patients was causing reduction in our 4E targets, such as MBS1 and cyclin D. We also expected from our molecular biology that 4E inhibition should lead to decreases in phospho-AKT, and we saw that. The other interesting and unexpected thing that we saw was that 4E itself was lowered, and we never saw that in any of our cell line experiments. And as I'll show you later, we did substantial resistance experiments where we grew resistant cells in ribavirin for over 200 days, and yet saw no change in total 4E levels. And so we believe that this is actually a population selection, where the ribavirin is actually getting most of the 4E sensitive cells, and now you're seeing cells that are not 4E sensitive. I should say that both patient 6 and patient 10 achieved blast responses, and so what you're seeing here is not the difference between normal and a tumorigenic. What you're seeing here is actually a difference in the blast population before and after treatment. What we can also see nicely is a reduction in RNA export, as you would expect by both inhibition and lowering of 4E, where we're looking here at this particular target MBS1, and at clinical response, you can see the patient had a substantial lowering of her RNA export activity. So that sort of provided support that we could target EIF4E activity with ribavirin in patients, and that this could correspond to clinical response. What I don't have time to show you, but is published in part in our blood paper, is that RNA and confocal analysis showed that non-responders had no effect on 4E activity. And when I go through the resistance section at the end, some of the reasons that that is true will be discussed. So this is showing you one of the things that happens at relapse. This is a patient who achieved a blast response. In red are bone marrow blast percentages, and in blue are peripheral blood responses. So you can see when the patient here at 56 days had her best response, we had a massive relocalization of 4E, very similar to what you saw for the patient I first showed you with a complete remission, when you compare that to the before treated cells. Unfortunately, what we note is that at relapse here, where we have blasts climbing very high, we also see a nuclear re-entry of 4E, indicating that probably we're no longer able to target 4E, and as I'll show you, that's true, that the ribavirin is getting chemically modified and no longer binds 4E in these relapse patients. So what to do next? Well, obviously we want to understand the molecular underpinnings of drug resistance, and we would want to try a traditional approach of combining chemotherapy with our targeted agent to see if we can improve duration and frequency of response. We chose low-dose Ara-C for this combination because there's been some evidence that, first of all, this drug is well tolerated in the over 60 population, versus high-dose chemotherapy, and secondly, some evidence that low-dose Ara-C by itself could have some single-agent activity. So we carried out a trial, a phase 1 trial, which we just completed, and we did run into a number of PK issues, but what I'd like to show you is some of the results we got for the patients who had reasonable amounts of PK given the reasonable amounts of ribavirin in their plasma given the dosages that they were getting. We see that this number should be, for a response, really needs to be about 20 micromolar or more. We had 10 patients that fell into this group so far. Of these, we had 2 complete remissions, so one of which was very long-lasting. This patient responded for 2 years, and that was particularly satisfying because she had a AML that was secondary to her breast cancer chemotherapy. And we had another patient who had a CR responding for 9 months. We also had a patient who had a partial remission for 4 months, but had a dose-limiting toxicity and had to come off of ribavirin. So although he was still clinically responding, he had to come off trial. We had 2 blast responses for 4 months, and we had 4 stable diseases, and unfortunately, we had a progressive disease. So one nice thing that we can see is the molecular targeting, and particularly at the confocal level, which allows us to measure, even at response, when you have very few cells to measure anything for, that we could see 4A relocalization continues to correlate well with clinical responses. So what you're seeing up here is is the percent of bone marrow blasts in patient 1, who was the one who achieved a response for 2 years, dropping steadily over the start of her treatment. And in the bottom, we can clearly see 4A confocal micrographs. And where you see the drops really starting to happen, we start to see really nice relocalization of 4E. Unfortunately, I don't have to show you here, but at relapse at 24 months, this patient had re-entry of 4E, very much the way I showed you for the previous patient with the blast response. So obviously, one of the key things that we would like to understand is the basis of ribavirin resistance, because perhaps that would allow us to get our patients to respond for longer, or for patients that are suffering from primary resistance to respond at all. So here is just showing you some of the biochemistry of ribavirin entry into the cell. So here is ribavirin. In order to enter, ribavirin has to go through the ENT1 transporter to cross into the cell. And then ribavirin has a very important step where it has to be phosphorylated to become ribavirin monophosphate, and this is through adenosine kinase. And then multiple kinases can act for the ultimate conversion of ribavirin to ribavirin triphosphate, which is its active form. What we found when we made cell lines is that many of the cell lines had a loss of adenosine kinase, and this was a major reason, at least in cell lines, for ribavirin resistance in what we refer to type 1 ribavirin resistance, which is ribavirin resistance where we see a net uptake impairment as measured by tritiated ribavirin uptake studies. So mutation of this and also the nucleoside transporter really prevent the net uptake of ribavirin. So we went and we looked to see how often this kind of resistance could potentially help us understand how patients were becoming resistant. We looked at one of the CR patients from a ribavirin monotherapy trial and a patient that never responded in the ribavirin monotherapy trial. And we used real-time PCR during the course of treatment, before treatment, during response, and end of treatment. And what you can see, for instance, in patient 11, is that this particular individual had a very large drop in ADK levels, as you can see here, and also in the transporter itself, indicating that this patient was probably resistant because no ribavirin was actually entering the cells. Patient 9 is a patient who never had a molecular or clinical response. And what's interesting here in dark blue is normal ADK levels. And in blue and in pink are before treatment and at 28 days for this patient. And this non-responding patient, you can clearly see, has about 5-fold less ADK than a normal person, strongly suggesting that this patient also is resistant because the ribavirin really never entered the cells. Now we have a second type of resistance that we refer to as type 2. And this is distinct in that you have normal drug uptake, but you have a loss of the EIF4E ribavirin interaction and no detectable mutation in EIF4E, nor are there changes in EIF4E levels. So let me show you the 4E ribavirin interaction data. So what we have here is 3 cell lines: a parental cell line, our type 2 resistant cell line, and our type 1 resistant cell line. The type 1 resistant cell line is the one with the ADK mutation. No ribavirin is taken up, as you can see here in pink, and thus there's no EIF4E ribavirin immunoprecipitation. However, you can see here with the type 2 resistant cells is that ribavirin uptake is normal. It's indistinguishable from the parental cells. However, there's no ribavirin 4E interaction in these cells relative to the parental controls. So we obviously wanted to know why that was, and all EIF4E activity was normal. There didn't seem to, the cells, the type 2 resistant cells were still sensitive to siRNA to 4E, indicating that 4E sensitivity was intact. 4E bound the cap to the extent, and in general was completely normal in these cells. So we wondered what causes this type 2 resistance. And in order to address this, we used deep sequencing methods, and we observed a 17-fold increase in the sonic hedgehog signaling pathway transcription factor called GLI1. And this elevation is specific to the type 2 resistant cells. As you can see here, our type 2 resistant cells have high GLI1. The cells that have the ABK phenotype are the same as parental controls. The other things that we could see, or follow on to show you, is that GLI1 overexpression in sensitive cells is actually sufficient to impart ribavirin resistance and also resistance to other drugs, as I'll show you. GLI1 RNA-mediated knockdown led to reversion of resistance. And this led us to investigate, and I'll show you, that the GLI1 inhibitor that's recently been approved by the FDA for metastatic basal cell carcinoma, known as GDC0449 or Vismogib, reverts resistance in these cells. And we went on and checked patients in a monotherapy and also in our combination study, and have been able to show they have elevated GLI1 upon resistance relative to clinical response. So this is just showing you some of our primary patient specimen. Here you can see GLI1 elevation of RNA levels before treatment, during response, and at end of treatment. And this is a log scale, so you can clearly see that we have substantial increases in GLI1 RNA consistent with what we saw in deep sequencing. And we also see here that if we look at a patient who achieved complete remission at response versus relapse, that we have substantial elevation in GLI1 levels, as you can see by confocal microscopy. We chose confocal microscopy in that Western blot here is because at response we have so few cells to work with, about 100,000, that there weren't really many other options. What we can also see is that non-responding patients also can have very elevated GLI1, perhaps suggesting a basis for why those patients who have normal ADK and are yet resistant to ribavirin upfront. So we went on to look at the ability of GLI1 to modulate the 4E ribavirin interaction. And again, we used our tritiated ribavirin immunoprecipitation experiment. Here's the IgG controls. And what you can see here with the 4EIP in either the flagged vector control cells, in pink, the sensitive cells overexpressing GLI1, or in blue, the type 2 resistant cells that I've already shown you. And what's striking is simply GLI1 overexpression by itself is sufficient to lose the interaction relative to the vector control cell lines. So GLI1 overexpression also imparts resistance to ribavirin. We can see the parental cells at their IC50 relative to now the cells overexpressing GLI1, or the resistant cells that were derived by long-term growth in ribavirin. So how could GLI1 be causing drug resistance? Well, our deep sequencing studies also suggested that we look at the UGT glucuronol transferase family of enzymes, which transfer sugar moieties onto metabolites and xenobiotics and can alter their activity in this way. And so what you're looking at here is a Western blot looking at either parental cells, the type 1 resistant cells that have impaired drug uptake, or the type 2 resistant cells. Again, high GLI1 and high UGT1A. I should say that the UGT1A family shares a common, they have the same molecular weight range, and they share the common carboxy terminus, and the antibody we're using here is a pan-UGT1 antibody. So we're seeing all the different forms of UGT1 together. So globally, what this suggested to us is that if you elevate GLI1, this causes an elevation of UGT1A, and this will eventually lead to a form of ribavirin that's being glucuronidated that can no longer bind 4E, and this underpins our resistance. Just to show you that UGTs can also be elevated in our patients, like the GLI1 was, we can see a patient, the non-responding patient. Again, they have very high GLI1, very high UGT relative to a patient in response, and a patient in response relative to end of treatment when they've relapsed has an elevation of UGT1As. Again, indicating that the UGT1A, like GLI1, is being elevated at relapse, or perhaps is elevated as causes primary resistance. We show here that UGT1A levels correlate with GLI1. For instance, if we SI GLI1 here, you also have a reduction in UGT with no concomitant change in 4E. Again, if we overexpress GLI1 in these two flag cells that you saw some of the biochemistry already of, you can see that you have a substantial elevation of GLI1, sort of commiserate with the same level of UGT1A overexpression as is observed when you have the resistant cells themselves. So UGT1A levels correlate with GLI1 expression. So obviously, though, it doesn't mean that the UGT has actually glucuronidated ribavirin, and so we used mass spectrometry in combination with hydrophilic chromatography to identify ribavirin glucuronides. So if we took either the type 2 resistant cells or the parental cells and looked, we saw the metabolite, for instance, ribavirin diphosphate. And in the resistant cells, we also saw the ribavirin glucuronide. And we can establish that it's glucuronidated and the carboxyamide of the triazyl ring using the collision-induced fragmentation techniques of MSMS. What you'll notice is that the same peak is not present in the parental cells, indicating that this is specific to the resistant cells. What we can see here is asking the question, well, if ribavirin is glucuronidated, does it indeed affect the ability of 4E to bind ribavirin, to bind it? And so in this experiment, we've taken purified EIF4E GST, and we've taken pure ribavirin, RTP, GTP, and we purified ribavirin glucuronide. And what we can see clearly is that in this competition assay is that 4E binds ribavirin in the presence of the buffer blank, but it can be easily outcompeted by either ribavirin or RTP, but is not competed by the negative control GTP, and is not competed for in this tritiated ribavirin assay by ribavirin glucuronide. And importantly, GST is not binding tritiated ribavirin by itself. So what we can say from these studies is that the EIF4E binds ribavirin, but not the ribavirin glucuronide. So obviously, we'd like to establish pharmacological means to control this. And what you're seeing here is just looking to see whether addition of the GLI1 inhibitor GDC could allow resensitization and loss of the glucuronide. So here, let's first look at resensitization. So in light blue are the type 2 resistant cells, and in dark blue are the parental cells. And what we can see here is that if we add ribavirin at the IC50, the parental cell responds, whereas the resistant cell does not, as expected, given it's a resistant cell. But if we add GDC and ribavirin together, you have a nice cooperative effect where the resistant cells are now responding. And I note that GDC by itself does not have any marked effect on either the resistant or the parental cells. So GDC is one inhibitor. It inhibits by binding the extracellular receptor for GLI1 signaling smooth-ins. We also wanted to take a second approach to check that we saw the same effects. So we used a drug GANT61, which directly binds the GLI1 inhibitor, and we see the same effect, where GANT61 by itself doesn't affect growth, but GANT61 in combination with ribavirin affects both. You can see both parental and the resistant cells. So does this pharmacological inhibition correlate with changes in glucuronidation? And so obviously we started looking at levels of UGT1A. So you're looking at parental cells versus the 10R cells. And what you can see is when you add GDC, you substantially lower GLI1 levels, which has been previously established to be part of the mechanism of GDC action. And this correlates with a reduction in UGT1A. So this is a very clear correlation. And if we take these same cells and we check to see if 4E can bind the ribavirin, use our tritiated ribavirin IP technique, you can see in the DMSO controls that the resistant cells don't bind 4E does not bind ribavirin, and the parental cells do. However, in the cells that have been treated with GDC, now you've restored the ability of 4E to bind ribavirin. And if we look at the mass spec data, we see that in cells that have been treated with the GDC inhibitor, we no longer see the glucuronidated form, which occurs at about 421, and we see the main peak being ribavirin diphosphate. So this indicates that pharmacological inhibition of GLI1 represses UGT1 and restores ribavirin for interaction by eliminating the ribavirin glucuronide. GLI1 overexpression alone correlates with the formation of ribavirin glucuronide. So if we take either FADU-GLI1 parental cells or the flag control, you can see that the glucuronide is only present in the cells that overexpress GLI1 and not in the cells that are only express vector. And as you'd expect, if you overexpress GLI1, you lose the ability of 4E to interact with tritiated ribavirin, once again showing that the ribavirin glucuronides can be formed by GLI1 overexpression, and this is through UGT1A overexpression. So because glucuronidation is such a broad metabolic effect on many drugs, we asked the question, could we have a multi-drug resistance mechanism occurring? And so we looked at whether GLI1-dependent drug glucuronidation could also impart ARC resistance. And I remind you that ARC is the cornerstone of AML therapy, and so could have wide-ranging sort of implications. What you can see in this graph is looking at either cells treated with DMSO or cells treated with GDC, and we have either the type 2 resistance or the parental cells. And what's clear here is that the resistant cells obviously are resistant to also ARC, despite the fact that the resistant cells had never seen ARC in our assay, so that was interesting. But what was also very interesting was that only the combination, not GDC alone, but the combination of GDC and ARC or ribavirin allowed drug sensitivity once more. And we saw a similar story for GLI1 overexpression, where again the GLI1 overexpression led to to ARC as well as ribavirin resistance. And finally, if we do SI to SI GLI1, we get a similar effect as we do with GDC, where once again SI to GLI1 allows restoration of ARC sensitivity to our resistant cell line. Finally, we asked this question whether this could be relevant in primary human AML specimens that we obtained from several tissue banks. And what you're seeing in here is that the blue colored in this colony formation assay, you can see normals. We have 2 different normals, and we also have 3 different AML patients. All of these AML patients were diagnosed as M4, M5 AML, and in all cases they were drug resistant. They'd failed refractory or relapse specimens. So a very hard group to treat. And we see that when you treat these cells with ribavirin or 200 nanomolar ARC, you don't really see very much of an effect. If you treat them with GDC alone, you don't see very much of an effect. But if you treat with either 200 nanomolar GDC or 1 micromolar GDC, you can clearly see that all of the AML specimens are sensitive in combination with ribavirin or ARC. So this is very interesting. The GDC alone is not having an effect. And I should note that all the drug concentrations we're using here are easily clinically achievable. GDC is found even 10, 20 micromolar in plasma. So these are physiologically relevant concentrations. So what this shows us is that GLI1 inhibition can lead to responses in resistant primary human AML specimens, at least in colony growth assays. So what's our model? Well, this is roughly our model for drug resistance. It starts off, at least in this form of drug resistance, with elevated GLI1. Elevated GLI1 leads to elevated UGT, which allows you to have glucuronidated ribavirin form. And this causes a loss of the ribavirin 4E interaction, which allows enhanced growth and drug resistance. If we use any of the strategies to inhibit GLI1, either RNAi, GDC0449 or vismodegib, or the direct transcriptional binder of the factor GAN61 that directly binds the GLI1 protein, all of these will cause a reduction in UGT1 levels. We no longer see the ribavirin glucuronide. We have restored targeting of ribavirin to 4E, and now we have growth impairment. So I'd like to just conclude. Hopefully today I've shown you that targeting 4E in a phase 2 monotherapy trial with ribavirin shows remissions and reasons for hope. Hopefully I've shown you that 4E is an interesting molecule and it works well beyond what we think of normally in the realm of translation. I've also shown you data from a second ribavirin trial, which is just finishing up, showing that we have nice responses with low-dose Ara-C and ribavirin. I've shown you a new mode of drug resistance via GLI1-dependent glucuronidation, and this is certainly going to be an area that we would like very much to follow up clinically with the hope that one day we could combine GLI1 inhibitors with glucuronidation to potentially inhibit glucuronidation in patients. And it also brings a general question, which I think is that maybe we should be looking for drug resistance earlier on in patients and not just decide that our drugs are failing after a month, but perhaps they worked for a bit and now the cell has outsmarted us and we need to figure out how these resistance mechanisms work. Before I take questions, I'd just like to acknowledge many of the people who did the work. I have to thank the clinical trial research unit and all my clinical colleagues, particularly Sarit Asseline and Wilson Miller. I have to thank Andrea, James, and Steve at PharmaScience for all the help they did because they helped us complete the mass spec. Hiba was the main runner with the resistance project. I think she's done an awesome job. Laurent and Mike did the lovely NMR data you saw. Biljana Klitschkovich did a lot of the molecular biology and the patient specimens and also did beautiful work on understanding how RNA export works. And I'd like to thank the rest of the lab, patients and their families for taking a chance with us, and of course all the funding agencies. And I've been asked before I do questions just to simply remind you that you can get continuing education credits if you go to this website. So now I will open up for questions, and unfortunately my site has completely won't let me look at any of the questions, so I will try to figure out what to do to get the questions. Okay, I have okay, I can only read, unfortunately, half the question. I have two interrelated questions. Let's see if I can. Unfortunately, I mean, maybe Tracy, you can email me the question, but my web interface is completely messed up and won't let me actually read the question. I think one question says the therapeutic potential of inhibiting 4E and GLI1 in other cancers, and I think that's a really cool idea. Right now we're trying to get a lot of microarray data, TMA data together to really try to look through and see how often these cancers have GLI1 and UGT at relapse, and especially trying to find some array data that has these guys happening together. Anything with 4E, GLI1, and UGT, I think would be really great potential targets for GLI1 mediated drug resensitization. So I don't know, and unfortunately I really can't manage to look at the other question. I know it says questions in the chat box, but my screen is actually freezed into this half. I can only see a little bit of the question questions. I can't even see the chat box at this point. And I'm afraid if I reload, I'm going to actually go off the air. But so I'm really sorry for that. But in terms of whether 4E can exist extracellularly in body fluid, I'm not really sure if anyone's looked at that. It's typically considered to be a cellular protein. I do note that glucuronidation itself is known not only to cause increases in drug solubility, and thus the idea that drugs can be more readily exported, but oftentimes drugs activity simply changes, and so the glucuronidation causes can cause proteins to bind new factors that they didn't bind originally. Some very interesting older work, testosterone, shows that sort of thing. So I think we should be sure to remember that glucuronidation doesn't necessarily mean increased export, just means a change in function. And in fact, although it's typically defined as a detoxification pathway, it can actually lead to increased drug toxicity even. So I think with that and my inability to really read more than half of the question because the screen froze, I'm really sorry. Please email me and I'll be really happy to answer your questions then. Thanks very much.